you are acquainted with the IPOPI website, you will know that we've had for a number of years a guide to the safety and efficacy of immunoglobulin therapies, which I think is a really great document in particular for regulatory advocacy in developing world regions. And Albert had kindly worked on that a few years ago. He had done the same for the World Federation of Hemophilia, and he has just updated that guide. And so the guide will be launched online on the IPOPI website shortly. We're also in the process of designing the guide to make it look pretty, and it will be circulated widely to the whole IPOPI membership. So I will now introduce Albert. I think many of you know Albert. He's somewhat of a regulatory guru in the field of plasma-derived medicinal products, and he has worked for many years for the regulatory agency in Australia. And Albert has then held various positions with industry and on a consultancy basis. And obviously he's one of the experts on the matter that will be discussed on this session. So I'm delighted to welcome Albert to the stage.
This is not the presentation. I need the first one, which I gave them. This is the second one. The one in the. This is the one. Okay, that's it. Yeah. Okay. All right. So let me see if I can do this myself. Okay, thank you very much. Good morning, possums. For those of you who are acquainted with one of my countries of origin, this is a salutation from the southern hemisphere. It is entirely affectionate. I'm very happy always to interact with this group. Now, you will note from your program that it is specified that I'm supposed to be setting the scene. Now, the other member on this panel is my dear friend for many years and ex-boss, Jan Bult. I have not enough courage to set the scene for somebody like Jan Bult. So what I'm going to do in the next few minutes, in order to, in fact, give some background to the whole area of the therapies which you utilize, is to make some comments and some extracts from this guide, which, as Johan said, has just been updated, and I rejoice to hear him say that it's about to be also prettified. I had no idea it was needing prettification, if that's the word, but I'm very happy to have it prettified. I'm actually feeling way ahead of the game this morning. Magda, about half an hour ago, told me I don't look my age. Johan has designated me as a guru, so I feel I'm winning. I really do. Okay, as Johan said, this guide was actually drawn up around about 2010 and 2011 in my spare time. And since then, a number of issues have arisen which do affect the immunoglobulin aspect in terms of safety and quality, and also, I think, in terms of the reimbursement and access aspects. So it was quite appropriate when I was asked to update the guide that this should be the case. This slide simply shows what I think are the current and past covers of the book, and I don't want to guess what the pretty version will be like. You all know that there are now a substantial number of immunoglobulin products on the market. Many of them are represented in this ESID Congress, and the lavishness of the booths of the various companies will obviously indicate to you, if you needed any indication, that this is an extremely lucrative product. And later on this morning, I shall intimate to you how lucrative it is for this industry. But there are a number of products. They differ now in terms of the methods of manufacture. This is an important issue, but what underpins this is our recognition which I reiterate every time, that there's no such thing as a generic immunoglobulin product, at least in terms of safety aspects, because we know that the different products behave differently in the patients, and so we are resistant, without debate, and appropriate modification to the concept of biosimilarity in these important products. Since the first edition was published, people would say, well, they were around even then, but I think we have had a consolidation and a much more higher level of visibility in different formulations in these products, and I allude specifically to the growing surge in subcutaneous presentation. This is now very well established in a number of patient groups, and it is increasing in terms of its importance. Not all the companies have it, but they are all striving for it. There are many clinical studies underway, and one particular company has kind of introduced, I was going to say, a sexy dimension to this, but following Johan Prévost, I will say a pretty dimension to this in terms of increasing the subcutaneous space through enzyme treatment in the hope that this will increase access once presented to the patients. This slide shows the different pharmacokinetic profiles. I think you know this very well from these different types of products, and the point to be made is that different patients will benefit to different aspects in relation to these formulations depending on their particular clinical needs. And I think that this allows me to immediately launch into what I hope is my primary message from this talk, which is, again, in the past 6 to 8 years, we have had a great emphasis on the so-called personalization of treatment in all aspects of therapeutics, including in immunoglobulin aspects. And this is a slide. The first time I showed this slide in public, I got into deep trouble. It was actually at an ESID, a small ESID meeting some years ago in Freiburg, and a very dear colleague, I don't know if she's in the room at the moment, I suspect not, took umbrage with my interpretation of the slide. But this slide comes from a little publication in the United States. And it shows the big danger of attempting to generalize here, not the treatment itself, but the delivery in terms of reimbursement. And what this slide shows is these are two different patients. And that shaded bar, that shaded space in the slide, shows a therapeutic course based upon the guideline-driven dosages of virtually every single professional association, as well as the product specifications in the product inserts. And the point to be made is that both these individual patients suffered greatly by being treated like this, because these bands of dosages were not adapted to their particular needs. Their actual biological levels of trough-based IgG delivery were different, and so until the patients started to be treated on the basis of their particular personalized needs, they had to suffer infections. And this is a deplorable state of affairs, because what is shown in this slide is actually a reality that some of the reimbursement authorities, particularly in the insurance in the United States, insist on patients having to be first trialed on the so-called guideline evidence-based driven dosages until they get sick, and then they have to be changed, which I think is not what we want. Fortunately, things are changing. I'm happy now to very quickly show some slides from my very dear friend and collaborator, Isabella Quinti in Rome, who has worked a lot in developing personalized algorithms based upon phenotypic and genotypic characterization of patients. Incidentally, I know that I'm hurrying too much. Anybody who wants these slides can have them. It's not a problem. They're on the computer here. Just help yourselves. This is all public information. But Isabella, amongst others, has shown that by attempting to characterize further patients in relation to their personal particular needs as expressed in their clinical profile, we can get better therapeutic courses. And for example, in this very nice study in which dosages to patients were adjusted on the basis of these profiles, she showed that actually those dosages can be adjusted and, in fact, in many instances, also lowered in relation to what the patient needs. So these are messages which are important for promulgation, even to the reimbursement authorities, because they can show that it is not just a case of increasing the cost of treatment. By personalizing, we can actually, in the first of all, ensure good medicine, but also ensure good economics. I want to say a couple of words on adverse events which have become more visible since the first edition of my guide. And you all know that, in fact, pretty much at the same time as it was being published, we had this great furor in relation to thrombotic events in the community of IG users. This slide, which is, I think, extracted from the journal Transfusion from the FDA's survey, shows that this was a problem actually associated with more than one product. It is very important to note this, okay? Because this was, while not exactly a general problem throughout the industry, it was a problem in more than one product. It was for this reason that the PPTA, which I worked in at the time, collaborated with the FDA in a very important workshop which happened in Washington. I think it was 2012. Yeah, I can't remember. And the proceedings were actually published, and this slide is from those proceedings. Since then, the industry has modified the methods of manufacture and undertaken other measures to lessen the risk of this adverse event. I have to remind you that thrombotic complications were recognized as a rare adverse event for IVIG before we had this big problem in 2010 to 2011. So this is always lurking in the background, and we need to be aware. Another problem, which also elicited a joint seminar in Washington between the PPTA and the FDA, is the issue of hemolysis. And again, this is a historically acknowledged rare adverse event which was well characterized but was very, very rare. Well, all of a sudden, we became aware that some of the product types on the market were, thanks to the pharmacovigilance systems which we have now, exhibiting higher rates than expected, still low rates, but higher than expected, of hemolysis in patients. And it was very interesting, as a result of efforts made by industry and the regulators, to note that this was actually characterizable in terms of the product manufacturing method, in the sense that products which were made by the historically embedded cone fractionation system, which I showed amongst other things in one of the first slides, did not have anywhere like the incidence of these events as the more modern products manufactured with modifications of this system and which were modified, in fact, to allow higher yields, which are important in terms of the necessarily high supply, and also more concentrated formulations. As you well know now, 10% has become an industry standard, and also higher concentrations in the subcutaneous. Well, these deviations from Cohn's venerated chemistry certainly led to a higher concentration of isoagglutinins, which led to hemolysis. Again, the industry took this on board, modified the methods of manufacture, and these rates now are back to the historically very low levels. But this leads us to reflect and take lessons. I will round off my comments today with a couple of comments on what I've already hinted at, which is the reimbursement space. I am, as Johan has kindly intimated, somewhat experienced in matters of regulation. Safety, quality, and efficacy are the historical barriers which a company seeking to acquire marketing approval has to overcome. Nowadays, we look on the fourth barrier as being much more formidable in terms of reimbursement through cost-effectiveness demonstration. And because of this, we have had an explosion in the past 10 years in the related discipline of assessment of health-related quality of life in the immunoglobulin space, as well as with other areas of therapeutics, through elicitation from the patients themselves as to how they feel about their health. I am very, I would say, enamored of one of these exercises, which comes from IPOPI. This is from the global IPOPI survey. And I am very intrigued, as Johan and Josie and others know, when these data were first available, we discussed them. I note with interest that in countries where there is obviously historically a much lower consumption because of financial economic issues of IG than in other countries such as the United States and other European European countries. I was never going to say other European countries, which is a very bad thing to say. But you note that there is absolutely no relationship. And in fact, in the countries of South America, and I remember visiting Roberta and discussing this in Buenos Aires some 5 years ago, there is a high level of quality of life elicited. Another issue which I think we need to take notice of is the question of formulation. This is from the IDF, who, as you know, produces very beautiful and extremely useful surveys on a regular basis. This is, again, health-related quality of life assessment. Amongst the other outcomes from this assessment, there is no difference in this particular exercise in patients' perceptions between intravenous and subcutaneous immunoglobulin. And I think these are very important data sets because they are actually the kind of things which feed into the reimburser's decision-making in terms of ultimately setting access and price issues. So these exercises are extremely important, and we must take note of them. So if I can round off on this setting of the scene, which I don't know, it can be classified as such, I would say that in order to have good principles in choosing and paying for immunoglobulin products, we have to recognize that, of course, these have increased and enhanced the lives of the patient community. The number of these products has increased, and also we have had differences in concentration, infusion speed. I haven't talked about that. All the products which are on the market through the approval processes of the United States, Australia, and the European Union are safe and effective. This is, if you like, a rubber stamp which is to be shown great confidence. Adverse events have occurred, but we have to recognize that these have come about because of the pressures in supply modifying the methods of manufacture, and therefore we need to be very careful when we rock the boat. And so have I, yes, indeed. And so I have been told that I've come to the end, and I have indeed. So I thank you for your attention. Now, I don't know how we're going to go on with this. Who speaks next? Jan?
So I'm coming. I'm coming to the rescue.
Good. Rescue.
Okay. So I'm not going to invite.
We need to pay attention.
Could we have one more question I'd like to answer to Albert, please? Because you said there are low numbers and the cost of therapies will be higher. And I believe you also have indicated that the price will go even higher still in the future.
Well, first of all, you will notice that in my slides, I actually had another short presentation about this, but that's okay. And you will notice that in my slides, there is absolutely no affiliation designated. So what I'm about to say is purely my own view. I always make this point to stay out of trouble. It never works, but I have to try. Okay, so if you look at the immunoglobulin space, it's actually very interesting at the moment. You know very well that your community only absorbs in the high-priced markets about 1/3 of this product. Much of the rest of the immunoglobulin production is designated towards a wide range of autoimmune disorders, many of them neurological in etiology, which absorb large amounts of immunoglobulin on a basically immunosuppressive mode of action. Now, the thing is this. At the moment, there are large numbers of potential, what we call disruptive therapies in relation to this particular area. There are a number of interesting biotechnological drugs being developed, many of them, I hasten to add, in conjunction with the main plasma producers, such as the big three, CSL, Shire, and Grifols. And these have the potential to substitute in potentially medically beneficial ways for the current therapeutic paradigm. So what does this mean? This means that if we look into the future, if these treatments really come online, if they do present an economic as well as a clinical challenge, you will see that more and more of the allocation of costs, which Jan-Bolt is correctly specified, will go on, in fact, the immunodeficiency space. So, again, my opinion, and nobody else's, although actually there are other people who have this opinion, is that if we do see a shift towards this kind of biotechnological application to the neurological space, then the costs will have to be reallocated more and more to the PID type space, and the